Jain friends, in part 1 we have learned about some of the terminologies. In part 2 we have discussed the importance of Mr. Mendel and his experiments. Now we are going to discuss about experiments which were being performed by Gregor John Mendel. The very first experiment, that is known as monohybrid cross. Monohybrid cross. As the term mono represents, we have already discussed in part 1 that as per the counting of science, mono means one. It means these were the experiments in which inheritance of only one character sorry, was studied by Mr. Gregor John Mendel. It was one of the finest decision by Mr. Mendel again his success, because he very first of all studied inheritance of single character, and then he complex his experiments furthermore. Now what he do, he performed the experimentation between male and a female plant. Male plant was tall, it was a tall plant, and the plant from which female gametes were received, that was a homozygous dwarf plant. We have already discussed about homo and heterozygosity. It is the situation in which both the alleles of the gene represents the character in same way, that is known as homozygosity, as per right here. When gamete formation takes place, only one allele of the gene transferred to the gamete. This is how a single kind of gametes get formed by the male, which possesses the character of height as tallness. Over here, also single kind of gametes get formed, which were having the character of height as dwarfness. Mendel performed cross pollination between two of them. He performed cross pollination, and as the result of cross pollination, the generation which get produced was having one allele of tallness while another of dwarfness. Surprisingly, all plants were tall. Here, the progeny from which gametes get formed, that is known as parent progeny, and the first progeny which occurs after experimentation, that is known as filial one progeny. That is filial one progeny. Now, when Mendel extended his experiments furthermore, then what he performed? He performed self-pollination. Self-pollination between plants of F1 generation. What happens over there? When gamete formation takes place for the plants of F1 generation, then it is possible that the allele which represent tallness reaches to the gamete. It is also possible that the allele of dwarfness reaches to the gamete, because because the self pollination was being performed within the plants. That's why the gametes, the we may say the composition of gametes of male will be same as per the female gametes. This is how same kind of gametes get formed. To understand this cross, a scientist whose name was Punnett, he gave a chart and a methodology. That methodology is known as Punnett chart. In Punnett chart, we draw some of the boxes We cross the male gamete from the female gamete. When this male gamete fused with this female, then the situation comes like this. If this male gamete get fused with this one, then the situation comes like this. When this gamete get fused with this one, situation comes capital T small t. And if this gamete get fused with this one, then the situation comes small t small t. Here Mendel observed that this plant was tall, this one was also tall, this one was again tall, and surprisingly, one out of four plant was dwarf in height. This is how Mendel reaches to a conclusion that out of these two kind of alleles, one allele is there which express itself in heterozygous condition. As we have already discussed in part 1, this situation is homozygous situation and this one is heterozygous situation. Mendel says that the gene which is able to express itself in heterozygous condition, such gene is known as a dominant gene. Dominant gene. But there is a gene which is unable to express itself in heterozygous condition, but it expresses itself when it comes to homozygosity. Such gene is known as recessive gene. Recessive gene. Nowadays, we write the dominant gene in capital letters and the recessive gene in small letters. This phenomena or this thing was known as law of dominance. This is how this becomes very first law of genetics, which was coined during Mendelian experiments, that is law of dominance. What law of dominance states is it states that the dominant gene express itself in heterozygous condition, but the recessive gene is unable to express itself in heterozygous condition, but it expresses itself in homozygous condition. First thing. Second thing, it was the second generation in the experimentation, that's why it was termed as F2 progeny. F2 progeny. Okay, now an important task comes here. What is that? That is comparison. If we are talking about the F1 generation, then as per visibility, all the plants are tall. There is no other plant. That's why we may say that the ratio of the tall plants along with another plant is 1, or we may say 1 raised to 0. Such representation of ratio, how the plants are looks or appears, such is known as phenotype. Phenotype is basically the ratio between the organisms on the basis of their appearance. But if we talk about the genetic composition, then all plants are heterozygously present. Here, the ratio also becomes 1, or we may say 1 raised to 0. But if we are talking about the ratio of plants as per the genetic composition, such ratio is known as genotype. We may say that phenotype of genotype of F1 progeny in monohybrid cross becomes 1, both. But if we talk about the F2 generation and we talk about the phenotype, here we may observe that out of 4 plant, 1, 2 and 3 are tall, but fourth one is not. Then we may say 3 ratio, the fourth one is single. is 1. As per appearance, the ratio between the plants is 3 raised to 1. It is the phenotypic ratio of monohybrid cross for F2 generation. Now if we reaches to the genetic composition, then we need to represent it as genotype. What is genotype of F2 generation in monohybrid cross? That is capital T, capital T. It is the only plant of its own kind. We need to write one ratio. There are two plants which are heterozygous, capital T, small t, capital T, small t. We will write them two ratio. It is the only homozygous recessive plant, that would be 1. This is how the ratio between genotypic condition becomes 1 raised to 2 raised to 1. It is often being asked in some of the exams. That's why it becomes some important. This is how it is monohybrid cross, which was being performed by Mr. Gregor Johann Mendel. That's all for this. Thank you very much. Have a nice day. See you in the next class. If you are unable to understand anything, any terminology or any process, then do write in the comment section. And if you enjoy it and you are learning something, then listen and see it carefully and do share with your friends. Thank you very much. Have a nice day. Jai Hind.